fridge now. You can definitely see some ropey, pillowy sort of situations
And
up
pockets
here.
of sediment that are definitely not sea stars.
Yeah, no, definitely not sea stars.
Definitely not.
Oh man, if it was a sea star, we'd be collecting it.
Agreed. That's why I'm looking for sea stars and not finding them. I remember from my first really deep dive on Nautilus how excited everybody got over a sea star, and we ended up backing up the ship for it, and we got it.
Ooh,
Nice.
very nice.
That was a tough thing because we didn't see the request right away, and we'd already moved on.
Oh, it's
Oh.
really
like-
hard to go back.
But we did it. We had some, you know, the pilots are just amazing on this ship. So we had a few features that we could use as landmarks in the backup cameras.
That's incredible. Oh my gosh.
That's why if you're doing an out and back hike, you'll always turn around at the start of the hike and note a few landmarks, just so you know exactly where you are when you're on your way back.
Yeah. Very cool.
Well, and also, if you drop a pin, our tracking is really very good, and we can get back to most places that we mark.
Nice.
Without any problems. So, like, if, say, we dropped off a sampler and had to come back for it, it'd be easy. Well, should be easy to find based on our navigation.
Tells you how far the Dead Reckoning system has gotten over time.
Well, and our Sonardyne beacons are quite accurate.
Oh, nice.
Yeah, back in the day with the subs, they used to tie 5-gallon bucket lids to things so that they can find them in the sonar.
Oh, there you go.
There's still some out there, I'm sure,
Oh, wow.
marking the spot.
Ooh, it's another sponge.
Pink flamingos.
Lawn flamingos? Yes.
Yeah, I really like pink flamingos.
Yeah, they're classic.
Lawn ornaments? Yeah, that's great. I really liked their little stunt with they brought a spatula down and they found a flapjack octopus and they tried to flip the flapjack octopus.
God. Really?
Yeah, it was just for a cute picture. They didn't hurt the octopus, but
okay.
it was really adorable. And it's like, oh, I'm making flapjacks.
Wow.
Well, there's another shrimp. One shrimply friend.
Ooh.
Yeah, another brisingid with more legs this time off to the right.
Yeah.
Oh. It's so elegant.
Oh, that's a good picture.
It is.
Are you a Benthosismidae?
Look
I think so. You see how it doesn't have a very long rostrum right between the eyes? It's just really short.
Yeah.
So this is a shrimp in the family Benthosismidae.
Wow. Oh.
Wow.
Ooh. Gorgeous.
Somebody calling?
Oh.
Ah, okay.
Ah,
Gotcha.
I see.
Hi, you have reached EV Nautilus. We are currently unavailable to answer your call, but you can reach us through the public chat. Ask your questions there at nautiluslive.org.
Oh, it is a nice crinoid.
Ooh.
This one, I believe, is in the family Antodonidae.
Wow.
Look at those really long cirri, those little legs that are holding onto the rock.
Are these the ones that can swim away?
Oh yeah, this
Yeah.
one 100% would swim.
Oh my gosh. How gorgeous. And what kind of things do these eat again?
What do they eat?
Yeah, what do they eat?
Little bits.
Little bits.
Little bits.
I love that.
Yeah, whatever they can catch.
How cool. Do they kind of just bring their arms to their mouth
Yeah,
area?
their mouth is on the underside,
Oh,
like most echinoderms.
wow.
Yeah, well, the current acceleration over ridges, that's a
Yep.
thing.
A little bit of forcing. I call it topographic forcing or orographics in the atmospheric community. Now what's curious is with the current up here why we're not seeing a higher density of life. We're just not seeing much of anything. Now there's going to be fewer, it is going to be a lower density community in general at these depths compared to some of the other places that we dive and the depths that we typically dive at, but I was expecting to see a few more corals.
Yes.
So the water is very clear. Maybe that's something.
Maybe. Yeah, they could be signaling lower nutrient load.
I also think it's a function of depth here. We're in this weird depth in between. Can we zoom right there in the center of the screen? I think there's a
Yeah, right there.
Oh.
A couple of them.
thing.
Oh, what are those?
I think it's a Megamouth Tunicate.
Ooh.
And some sand.
That's max zoom there.
Or it's a sponge.
Wow.
Was there another one that you wanted to see?
I'm pretty sure it's a tunicate.
I think the other thing I pointed out was just sand. You're good.
Okay.
Because apparently I'm doing that today as well as dropping the dive plan.
Sediment or friend.
Or tunicate. Or sea star.
Or sea star, yeah.
I mean, the camouflage is just incredible. Like, I don't know if I would have been able to spot this tunicate.
It had a different color.
Yep.
It's all about, yeah, I'm good.
There's like a tiny, tiny little blowy thing right below that too.
A glowy thing.
Blowy, like it was floating in the wind.
Oh.
Or the current, not the wind.
Yeah. The sea wind.
I just finished that book. So good.
Oh, you finished The Name of the Wind?
Yeah, so I got wind on the brain.
Now you know the name of the wind.
I do.
Yeah, where was the thingy that you saw?
It was right under the tunicate.
Oh, okay.
Yeah.
So, are we in the same spot?
Yeah.
Basically, yeah.
It's very tiny.
Tiny. I don't
It's
see
yeah, it's like
anything.
Yep, I see it. Kind of on the lower left.
Oh, yeah, right in the, oop,
No, it's still under the tunicate.
right there.
Oh.
that.
It's just one polyp.
Yeah.
Yeah.
It's
Oh,
just
yeah.
one little polyp, octocoral polyp.
Good
Wow.
spotting.
Yeah, that
I
was-
know, oh my
That was
gosh.
I didn't even see
That's
that.
incredible.
I have it a little more close up to me than you all do.
Okay.
Oh my gosh.
True. Somebody over there got higher resolution eyeballs.
Enhanced.
Enhance image.
She has a very nice monitor over there, though.
Yeah, it's true.
Exactly, yeah.
See a single polyp.
And I still can't see half the things Megan sees, so if you had what I had here, oh my gosh.
I could see all of the
You could see it
things.
all.
Let's upgrade to those 4K eyeballs.
Yeah.
Oh man, we give you some 4K eyeballs, you'd be unstoppable.
Oh yeah, that would be great.
I mean, it looks like on some of these crusts we're seeing some traces of things that have been here.
Yeah, there were definitely lots of things here at one point in time.
Yes,
We got
There
another
might be more
stock.
things here eventually,
hopefully.
but you know, we're kind of meandering around in the dark.
Yep, with our little flashlight and our compass. And I am here for the equal opportunity science objective achievement. See some more of those crinoids
Oh.
along with that sponge. Oh,
There's something to the
look, a
bottom.
couple of 'em.
Yeah, there we go.
There's a pair of them. See, there's more things.
Yep.
And there's, looked like,
Brisingids really like high current areas.
Yeah.
So, you know, they're doing pretty well here.
more crinoid.
Yeah, we see them get quite big in some of the high current areas.
Great texture on these rocks.
Mm-hmm. Yeah, this is like textbook pillow basalt texture. It doesn't get a lot better than this.
And oh, I already passed it.
Yep, this is a pretty flat area, so it's going to be a very gradual incline for a hot minute. We're about halfway between waypoint 3 and waypoint 4. So for folks who've been following along since the beginning of the dive, the waypoints are at the beginning not really spaced out evenly. They do get a little more evenly spaced out for most of the rest of the dive.
How many waypoints?
So from waypoint 1 to waypoint 2, there's quite a bit of distance there, and we set that up to meet some specific sampling objectives for eDNA and just make sure we had some good markers in place to remind ourselves to hit the pumps when we needed to.
And for this dive, we have in total 14 waypoints.
Yeah, wow.
We don't know if
Longer
we're
dive.
Amber
going
was
to see all
asking.
of them, but we're going to try for it. Yeah.
Oh, and do we already do a transect? Let me see our notes.
We haven't done
No.
any transects yet, and I'm not seeing much in the way of documentable, like, like corals and stuff here. So if we see something though, that yeah, I'd like to try for a transect.
red arms peeking out.
Some weird crust morphology
Yeah.
there.
Yeah.
I'm like, what's going on with that?
It reminds me of those puzzle pieces that we've been doing.
Yeah.
Yeah.
Oh, look how flat it is up here.
Yeah.
I feel like you can see for really far away.
Yeah. That's probably another dead sponge stalk that we're coming up on, isn't it? A couple of them.
Yeah.
Yeah. I don't know if Annie's going to want to transect at that.
No.
I don't think she'd appreciate that very much.
But the transect for the rocks would be excellent.
I'd be happy with a transect of rocks, but I'm a geologist.
Yeah, I'm surprised we're not seeing many bamboo coral.
Agreed. Yeah.
Usually, like, they're sparse, but
But they're there.
they're there. We did collect one, so
Yeah.
we know that they're around here. I see that little white spot. Is that a Munidopsis?
This one?
Oh, yeah.
There.
Good spot on that.
Ooh. Oh, look at that. Whoa.
Yeah, there's a little Munidopsis.
Wow. This is the type of squat lobster.
This is a type of squat lobster,
Yeah.
that's right.
Oh. This is a very
I think it's
bright.
Munidopsis bairdi.
Oh.
So with that long upturned rostrum.
Mm-hmm.
What kind of jobs do these little friends play in the environment here?
Oh, they're scavengers as well. They walk around and they eat stuff. They like small crustaceans and polychaete worms and any other sort of small invertebrate they can consume.
Oh.
They're not particularly picky.
Yeah. Just help cleaning up. Oh, cool.
Yeah, there's not a lot of stuff suspended in the water column for them to feed on around here, so. Or
Take what
much
they
to
can
feed on
get.
in general, so it's pretty slim pickings. Take what you can get.
Yeah.
Yeah. There's a really thick sponge stalk that's just going out of view.
Yeah. More brisingids.
That's a big one.
Yeah,
big
look
one.
at that. That's huge.
Now there's something green in the center of the field of view, and I can't quite figure out what that is.
Green.
Yeah,
Ooh, where?
right in there.
Oh, yeah.
Yeah, it's just something that's sedimenting there.
Yeah, sediments. I don't think it's actually green. I just think it's a little bit
It
yellow
averages
and
to
the blue
green,
and
yeah.
color balance.
Yeah.
Translated kind of funny.
Yeah, it's a mixture of some white and dark fragments, and yeah, just with
Oh.
how things are balancing out on our screens, it averages to a green.
Kind of looks like lichen or something.
Yeah, kind of.
It's a small sediment pile of some coarser material, but it's not clear what it is.
Yeah.
So usually we're used to seeing those pockets of really fine, basically carbonate mud,
Yeah.
that pelagic sediment that builds up everywhere, so it just looks a little bit out of place there.
Mm-hmm.
Take a little peek at our brisingid.
It's very striking.
Yeah, they're very pretty.
Mm-hmm.
Such a great color.
Whoa. And is the mouth in the center, or it's at the bottom as well?
On
right?
the bottom.
On the bottom. Wow. Oh my gosh. So
Generally,
nice.
the butt is on the top with these things. The mouth is on the bottom.
So
That seems
that's-
pretty convenient.
So that's the butt.
And then underneath, there are some scale worms. Seems pretty convenient.
Oh no.
I'm gonna put myself on mute.
It's okay, man. You're good. Yeah, so if we're seeing some evidence of feeding traces here on the rocks too, maybe that's what the scale worms have been responsible for.
I don't know if the scale worms would make such large traces.
Okay.
Maybe it's those like limpet things that we
Maybe.
see, like those slit snails.
The ones that we're hoping to find a couple of.
Yeah.
Yeah. Yeah, we'll keep eyes sharp. I mean, those kinds of traces can stick around here on the seafloor for
Yeah,
quite
for
a
a really
while,
long
so
time.
we don't know if that's recent or not.
I mean, you know, they don't move fast, but they could definitely scoot around a corner.
Not a lot moves fast around here, including the
But you know, it could be like,
incrustation process.
it was like, you know, a month ago it was there.
Yeah.
Just
Okay, a
not
month
there.
ago, I would consider pretty rapid
That's pretty fast,
in this kind
right?
of setting. I'm also thinking in deep time too, and thinking about like ferromanganese crust.
Right.
Yeah,
Well, sometimes like
I'm
those
skewed.
predatory sea stars can spend years on a coral.
yeah.
Oh wow.
I mean, metabolically, it's pretty slow at this depth.
Yeah.
It's cold. There's not a lot of food, so you have to be very efficient.
Wow. Just makes me wonder, like, what is the experience, you know, of all the animals that live down here? Is that another dead sponge stalk? Here.
There's a fish up
Ooh.
ahead.
Oh, where is it? Can you
Fish
point it
on
out?
cinema cam.
Oh, cinema cam. Oh, we can't see it in
Oh
HD.
yeah, look at that.
Oh, well, it's sort of
It's on the
off
left
to
side.
the port side. Up. Oh, tilt up.
Yeah.
Oh. Oh wow.
So this is a eel in the family Synaphobranchidae, and this eel is Iliophis. So it is in the subfamily Iliophinae, and remember when I was talking to you about the dorsal fin origin?
Yeah.
This one's dorsal fin starts really far forward on the body, more forward than the anal fin.
Mm-hmm.
Oh, slow down. And
Oh, there's another
the
something.
pectoral fins are relatively small,
Yeah.
and you can see that the jaws are pretty much in line. They don't have they have like more of an overbite than an underbite.
Mm-hmm.
And you can see as we zoom in, this fish has sort of pores along the lateral line that are quite prominent. And that's how I can identify this as an Iliopus.
And would this specific kind of eel be kind of like a top predator in the area, or?
Yes, yeah.
Yeah.
These eels are definitely the top predators and scavengers in the area.
Yeah.
One of the larger animals that you'll see at these depths.
Wow. Looks so ghostly.
And the common name for this fish is arrow-tooth eel.
Oh.
So is it the fin morphology that's very diagnostic of this group of eels?
Yes.
Okay.
Yeah, particularly the dorsal and anal fin origins.
Yeah. Okay.
And then the pectoral fins are so much smaller in the Iliophinae compared to the Synaphobranchid.
Gotcha.
Wow. That's so neat.
Up is so much spicier.
Yeah, being able to pick out the patterns and these little details and morphological difference between these species makes it so much easier.
Yeah, and another key character are the tubercles, the little like nostril tubes.
Uh-huh.
They have really long ones in comparison to the Synaphobranchid.
Yeah, you're teaching me a lot about what to look for different kinds of animal ID. It's, yeah, I just haven't been trained in this, so I don't even know where to start right now. And
I learned all this from Bruce Mundy.
Oh yeah.
He's a Hawaii fish expert. And he would call in during the Okeanos dives and tell us all about the really cool fish we were seeing.
Mm-hmm. Yeah, I love that Okeanos does those call-ins.
Okay, yeah, we can carry on.
Yeah.
Chase that eel enough. It was a very cool eel.
Yeah.
Yeah, you learn a lot from those call-ins.
Oh, yeah.
Yeah, when I was shore teaming for the Okeanos expedition earlier this year.
Caulophacus.
I always loved it when
What's this fish?
when
What
Chris
is
Ma
that?
would call
Oh,
in.
it's so fast.
Oh, it's gone. It was a Bazazetus.
Okay.
It was so fast.
Yeah, I was really enjoying when Chris Ma was calling in
I
to
love
talk
Chris
about sea
Ma's.
stars. He's so good.
Brisingids are his favorite.
I can see why. They're pretty spectacular.
Mm-hmm. He always has a lot of really fantastic facts about sea stars and other echinoderms. And I use his blog, the Echino blog, for all sorts of IDs. It's really helpful. And sometimes I'm annotating and he's already wrote a blog about a specific animal. I was like, I remember this one.
That's amazing.
Oh, that's great.
So, yep, this is a Caulophacus sponge, a glass sponge, in the family Roselidae.
Yes.
Looks like a mushroom.
It looks like a mushroom. That's how you ID it.
Oh.
It doesn't taste like a mushroom, though.
Definitely not.
That.
There was another one a little bit further downhill on the other side of the ridge from where
I
we
saw
came
that
up.
one too.
Yeah.
Population quantity 2.
Yeah. If there's not much to look at up here, with how narrow this ridge is, we may have an opportunity at some point or another to drop over onto the starboard side of the ridge, and just take a peek down. You know, we don't have to go off to the side of it or anything.
It looks like the ridge here is now sort of nicely sloped down on either side. It's not as sharp as it was when we first got up here.
Yeah. Yeah, not much going on down that slope either.
Oh, there's 2 fish behind us.
Oh.
I can see it in the butt cam.
Yeah.
So coy.
They're more comfortable behind the camera than in front of it.
Yeah.
I get
Yeah.
that.
I can understand that.
Definitely. Right there with you, Amber.
We saw. We're moving on.
What's the little
There's
white
something
nubbin?
bright white, yeah.
I think it's a hold fast.
Okay. Just hoping for like a barnacle or something.
Yeah, no. It's
Yeah,
not
that's old
barnacle.
fast. That looks like mucus or something.
Yeah.
Ooh.
So it looks like a lot of the ferromanganese around here is just like broken apart, and it looks like it's an old wasting process.
What is a wasting process?
It's erosion,
Oh.
basically. Probably a pretty slow process here, but yeah, this is a pretty thin ridge, and if there's any, you know, failure of the slope below these areas, it's possible that you know, there might have been some erosion or some rocks that are weathered out from underneath it, and it's just kind of collapsing down over time. It's a little hard to tell what's going on, but from a bird's eye view, it's the best I can do.
Yeah.
Val, could you describe the process for ferromanganese being, you know, spread around?
The formation of ferromanganese.
Yes.
Sure. So ferromanganese crusts
There's a
are
big
found
something up ahead.
throughout the Pacific. Generally the older, well, you find them in the other ocean basins too, of course, but I'm very Pacific focused, so that's where my biases are. The older these seamounts are, the thicker the ferromanganese crusts tend to be on the substrate, just broadly speaking. And that's because ferromanganese crusts form as a chemosedimentary process, which means that they basically are formed by iron and magnesium oxides primarily depositing out of seawater. So these are metals that are dissolved in very small quantities in the ocean, and that just ends up depositing out onto surfaces. And ferromanganese it's not really picky about where it will deposit. Pretty much if there's a surface down here long enough, it will start forming. So think of it kind of like if you have a saltwater solution that has a ton of salt dissolved in it, if you, under the right conditions, have like a little, I don't know, put some small object or something in there, like a straw or whatever, if it's super saturated and under the right conditions, it will start depositing salt out of that solution, basically crusting it over. So that is sort of the same process happening here. It's just ferromanganese deposition is an extremely slow process. So it occurs on the order of, at most, we think a few millimeters every million years. So when you see ferromanganese crusts that are centimeters thick, that's a lot of time recorded in those crusts.
Oh, look, a Chrysogorgia.
What is
And
this?
a really fluffy Caulophacus.
Oh
That is
my
extremely
gosh.
fluffy.
It's like a lollipop.
That is an absolute unit.
It is.
Thanks for that description, Val.
That's
welcome.
fantastic.
Unit.
It's true. So is this a type of sponge or?
Yeah, it's a type of sponge. Same as the ones we saw, well, same genus as the one we saw before, but this one is, I would say, probably considerably older.
I don't think I've seen one that looks quite like this before.
Yeah, this one is different species, I think, from the one we were looking at earlier.
It's
yeah.
like a yellowish color,
Yeah.
right? Kind of yellowish.
But it's
It has a little brown on
quite
it.
fluffy.
Yeah.
Looks like a marshmallow.
Oh, it does.
Levi.
I mean, it won't kill you.
Don't need it.
It does have a little cotton candy appearance to it, not gonna lie.
I mean, it would be soft,
Yes,
to some degree.
to some degree. Yeah. I'd
Other
still
than
wear
the
gloves
nice
when handling it,
glass
though.
shards that would be all up in the business, but
Oh my goodness.
That's the kind of cotton candy you wear gloves when you handle,
That
Right.
definitely.
which we have plenty of in the lab.
I love the little bumpies
Interesting.
going
but
up.
look at that texture.
Yeah.
But that's the brown I'm seeing.
It looks
So,
all gloopy.
gotta love the zoom on this camera.
Yeah, it's
Yeah.
amazing.
It really clarifies things.
Look at that. And what do you call the webbing? What did you call it earlier?
The dictyonal framework.
Yeah. Oh wow. That's so cool.
So hexactinellids, or glass sponges, are made up of spicules, and all hexactinellids have what are called hexasters, which are the type of spicules that have 6 like spikes or rays.
Wow.
And then to the left, there was a coral, right?
Yeah, there is a Chrysogorgia over here.
Another one
Oh,
of decent
can't see it.
size. It's not an absolute unit Chrysogorgia, but it is pretty big.
It's a good size
yeah.
one.
It's a good size. It is a healthy size.
They're just two best friends chilling.
Yeah, just chilling forever
And they were
together
roommates.
on the top of this rock.
They were roommates.
And it does have a roommate. See, there's a little squat lobster up in the business.
Oh
Mhm.
yeah.
Just hanging out.
Just chilling.
Yeah, it's a Uroptychus squat lobster. We often see this association between the Chrysogorgia corals or golden corals and the squat lobster Uroptychus. We call them golden corals because the skeleton is a golden color.
Oh, nice.
Oh,
Not to be
wow.
confused with gold coral, which is a completely different animal altogether. It's a zoanthid that grows much shallower. Oh, there's another cool animal here.
So I'm
Right
seeing
here, zoom in.
sorry.
A smack zoom there.
Yeah. There's a tubularid hydrozoan on
Oh,
this
yeah, right here.
Chrysogorgia. Yeah,
Right.
that's the one.
Oh, whoa.
Sure enough.
So it's a single polyp hydrozoan that has this sort of unique body form. Looks like a little tube.
Mm-hmm.
Wow. Your eyes are incredible.
Many years of looking at these types of things will do that to you.
Yeah, it makes it a lot easier to spot the nuances and things. Really dig into the details.
Yeah.
It
So does
takes a
the
long
skeleton
time.
actually
It does.
like metallic of this one?
Yeah, it looks kind of metallic or golden color,
Wow. That is so neat.
which is also the same as the gold coral that I was talking about, which is a zoanthid that grows in shallower water, about 350 to 500-ish meters, and it grows on top of bamboo coral. But it's unique in the zoanthids in that it lays down a skeleton on top of the bamboo coral, which is a proteinaceous material that looks gold. And that's the same type of material that this skeleton is made out of
Gotcha.
for the
Okay.
Chrysogorgia that we're actually looking at.
Wow. What a beautiful skeleton.
So one question that comes up every now and again about Metallogorgia skeletons is the metallic color actually metal content in the somewhere in the skeleton or in this proteinaceous cover, or is that just how it's sort of iridescing, the luster that it takes on because of how the skeleton is built?
I think it's just an iridescence that it just naturally takes on. There's no actual metal in it.
Yeah, which is always
Moving
really
on.
captivating about that, because that
You okay?
yeah, it's difficult to pull off that metallic kind of luster with non-metals. You can do it in some instances, but yeah, it's interesting to see that in a very biogenic setting too. So we're just doing a pilot swap here for a moment, and then we'll get ourselves resettled and going again. So sitting in Herc now is Megan, our awesome navigator and annotator and biologist, and also trained ROV pilot.
So many hats.
And former co-lead scientist on previous Nautilus expeditions. So she's done a little bit of back row and front row.
I like getting to fly the ROV. It's fun.
I would love to try it someday. But not today.
How did you get your experience in ROV piloting, Megan?
I just kept asking until they let me do it.
I love that.
Yeah, that's sound advice for
But I did a lot of,
something.
you know, training and learning
Yeah.
about the vehicle before I ever touched the sticks.
Yeah.
Yeah, you have to.
So You know, they want you to get to know how the vehicle works and responds and how to fix it in some respects before people are like, okay, we'll trust you behind the wheel here.
Yeah.
Yeah.
Knowing
Flying
how
is not easy.
to maneuver everything around is important. Oh,
Yeah.
I'm gonna go up.
Yeah, because I was talking with Levi about this earlier in the day, and it's one of the things we don't think about a whole lot. We live most of our lives, honestly, in 2 dimensions. You know, we exist in a 3-dimensional world, but we spend so much time moving around on effectively a flat surface. Most of us can handle maneuvering in a more or less 2D plane, pseudo 2-dimensional.
Yeah.
But you add that z-axis, that 3rd dimension, where you're also having to manage your altitude. That's a whole different ballgame, adding that into your spatial awareness. So like driving a car versus flying a plane or an ROV are going to be completely different.
Yeah.
Yeah. So that training matters.
Want to zoom in?
Yep, going in.
Ooh.
We're looking at some more brittle stars on the sponges. That's what we've been seeing a lot of here.
We had one guest ask, why don't they see the brittle stars on the ground here? Like we've just been seeing them on the sponges.
Well, the sponge stock gets them up off the bottom, and so there's probably more feeding opportunities for them on top of the sponge rather than crawling around on the bottom.
Yeah.
All right.
For the multipuffer, I turned off pump number 2, and now we're starting with pump number 3.
Roger that.
Can you double click on the Herc icon to drop a point there? On HiPAC, can you double click on the Herc icon? There you go. That should, yeah, there you go. Thank you.
Okay.
Bridge, pilot. I was calling for the bridge. Oh, you're on the bridge. Oh, wow. Are you going to take my ship move?
One moment, okay, sorry.
Can we do a 30-meter move? 300. Yes.
Correct.
Okay, cool. I was like, what, Meraki, are you on the bridge? All right, cool, thank you.
I was very, I was like, whoa, hang on. That was amazing timing.
I know. Ooh, a Chrysogorgia.
Ooh.
Ooh, what? Oh, there it is. Oh my gosh.
There's just one, otherwise I wouldn't want to do a transect here. But also that involves dropping some points, and we gotta manage that, so.
Yeah, do you want me to do a little zoom on that, Megan?
Sure.
Yeah.
Going in.
I think there's another squat lobster hanging out in there.
Oh yeah, looks like something.
I could probably set down. You zoom out.
Come on wide.
We got some time.
How?
Yep, this dive is
I
the
like
longest dive
practicing
we've done.
flying it in though.
Oh yeah, definitely keep the
That's
skills sharp.
tricky.
Yeah, this dive is going to be a total of 24 hours instead of the 12 to 14 that we've been doing on all of the previous dives. This is the longest dive that has been performed in the Marshall Islands sanctuary, marine sanctuary. Why am I always having so much trouble recalling the word sanctuary? I don't know.
It's a tricky word.
It shouldn't be.
It has like the think, like the k, and then chuary.
It just must be in some little corner of my brain that I don't use that much. So there's something behind there too.
Ooh.
Which doesn't make sense. It's a perfectly usable word.
This is
Yeah.
fun.
But yeah, down on the substrate, a little bit behind, and port to the left of the coral, there was something behind it, maybe like a hydrozoan or something.
Hydrozoan?
Yeah. Right down here.
Down. Okay. Oh, I think it's a bryozoan. Good spot.
Ah, okay.
Yeah.
Oh.
Really small stuff.
Very small,
That's
yeah.
so tiny.
That's the thing about zooming in, you know, keep seeing more and more things. Not everything down here is super large, but there is a lot.
There is, yeah. Yeah, one of the rocks that came up from yesterday's dive, the biology team took 5 subsamples off of it before I even did anything with it.
Oh, wow,
Oh,
really?
5?
Yeah, yeah.
Oh, wow.
I had a lot of associates on it, and several different like distinctly different kinds of associates. A couple different sea stars, some green things that might have been anemones, we're not sure. Yeah, I think like a juvenile sponge or something, if I remember correctly.
Oh, how cool.
They had quite a bit going on.
That's why I always loved picking up rocks as a kid and looking what's underneath
Yeah.
to see what's there.
Mm-hmm. I mean, we're basically doing the same thing, just professionally.
Yeah.
Professional
like,
rock
what bugs
picking.
are under here?
Yeah.
I definitely had a couple of pet potato bugs as a kid.
I love that. My
Named
favorite
one
find.
Jessica.
Named one
That's
Jessica.
amazing.
That's amazing. Oh my gosh. I think my favorite under rock find was a mama gecko guarding her eggs. It was
Oh,
so
wow.
cool. I was
Oh,
like,
that
oh
is
my
cool.
gosh.
You
I
had
spent
it?
like
Alright.
5 minutes trying to figure out how to put the rock down gently. So that's really cool to know. So we're really the first 24-hour dive to
Yeah.
be done here.
Yeah.
That's so incredible. Oh.
24. Val, that was 245 for the multi-puffer?
Yes.
Okay, perfect. And we switched from 2 to 3.
Yes.
Are those little white things something, or is that sediment?
It's not behaving like a sediment pocket. You're asking about that, correct?
Yeah, there's
Oh,
like
yeah,
two.
It's a little thing.
It's a little thing.
Yeah, it looks like something.
It kind of looks like a gingerbread from here, but
Gingerbread.
don't jump out of your seat,
I think
Levi.
it's just a shiny part of the rock.
Oh. Oh yeah, it is. Oh well, it got me. Well.
Yeah.
Like that is a shiny part?
That is not a shiny
No.
part.
Oh,
That is
what
it's
is
a
that?
sponge.
Oh, it's
Yeah.
it's a sponge.
The beginning.
We were fooled again.
It's
Yeah.
a very odd-looking sponge, but
Yeah,
like
it's an
it.
encrusting demo sponge.
okay. I
Maybe
don't
that.
think we see that kind of morphology all that often, so.
Oh, we usually don't see it too much because we're not always zooming in, but they're there. And
Yeah.
they're more abundant in some places than others.
Yeah.
This one doesn't look the healthiest,
Oh.
so, you know.
Would this be
brown
considered a young
Doing
specimen
its
or
best.
older? How can we tell?
I don't know. I feel like I'm leading, like I'll see it better.
Is that like a lip?
I don't think it is anything. I think it's
Okay.
just the way the sediment's sitting in that crack.
Yeah.
Dang.
It's sort of like when you look at clouds and you start seeing stuff.
Yeah. That's the perfect analogy for that.
But that sponge looks like a butterfly.
It does a little bit.
I know for sure the sediment has tricked me several times.
Or shadows?
Yeah.
Yeah. More Caulophacus.
Ooh.
That literally looks like an enoki mushroom.
It does look
does.
like an
Oh, I
enoki
want some
mushroom.
enoki mushrooms.
Ooh, enoki mushrooms are so good.
Oh, like when they do the thing where you have the ultra thin sliced beef and it's all rolled up
Ooh, yes.
in the with the enoki in the middle.
That's perfect.
So good.
Or in some sukiyaki. I love that.
Oh yeah, some sukiyaki. That'd be good.
It has some sort of
neat.
associate on the stalk.
Yes.
about
I think
halfway
it's
up.
a
Oh,
little
what is
amphipod.
that? It's so tiny.
Tiny amphipod.
It's two best friends. Chill.
Yeah, you know.
And they were roommates.
Yeah.
Oh, fuck. Laugh.
No, here
Do
we go
it. You can laugh.
Don't mind me, I'm just laughing.
It's pretty good. We encourage that.
It's like I can hear you holding it in.
Calm down, calm down.
It's my birthday.
Calm down.
Sea star.
Oh, really?
about
yeah,
time.
there is
Oh,
a tiny
let's
sea
go.
star. We'll probably be sucking that up.
Hey, it's not a sediment pocket.
It
Let's
really
go.
is a star.
Not sediment.
All right, should we set up for sampling then?
We are good to go for sampling. We are not moving.
Ooh,
Okie dokie.
excellent.
Is it
Sea star spotted.
going
Highlighting.
down? Going down. Here's your porch. It is easier when you can see the porch.
Yeah. It's easier when you have a porch.
Nope.
Too soon. Too soon.
Too soon.
Oh, and there's a
And a
bamboo
coral.
here too. Look at that.
Okay. We might do a double. We'll
Ooh,
make a decision.
a double-double?
Yeah, let's look at the bamboo first.
A bang-bang?
Since it's right there, yeah.
Well, we did collect an unbranched bamboo earlier.
We did, yeah. It looks similar in
So-
color, but I know bamboos are tricky like that, so we'll
They
take a
are
close look.
tricky. It's good to
Ooh.
zoom in and see.
Yes.
Enhance image.
Going in.
I still can't believe that that thing was hanging on by the suction hose.
Yeah, that's nuts.
It was one of those moments where I just walked in to make sure that Fanny got some breakfast, and there was a situation, because out on the deck we were not fully aware of
That's cool.
said situation,
That's a nice one.
and
Nice
It was
fleshy
much-
volcano-y polyps.
it was much funnier when it was done.
I think it looks similar to the one that was collected.
Ooh.
That
I think
one
you're
was
right.
quite fleshy as well.
It was. Yeah, it had really large polyps.
Are the polyps kind of tucked in a little bit?
Yeah,
like I
they're
see
all
Yeah,
one.
they're
closed
retracted.
up.
Oh.
And there's just the one that's like halfway
It's like
closed.
saying hi, but like a peace
Yeah, it's
sign.
like
little
peace.
Yeah, yeah.
Yeah.
Yeah. I think we'll leave this one behind, but
Okay.
we're going to want that little tiny star.
All right, let's set up to get ourselves a sea star.
The sea stars at this depth are not as well known, and it likely could be a new species.
Ooh.
It's right at top of the screen, straight back from the bamboo.
Yeah, if we end up in a more sedimented area again, I know Steve's been mentioning wanting to look out for Relicanthus.
Yes.
Yeah.
But it has to be a small one.
Yes.
Oh.
I remember what happened when we slurped the big one a few years ago.
Yeah.
Oh boy.
And I like it when the pilots are happy.
Oh.
What happened with the big one?
It was
Stuck.
too big.
Too big. Not good.
It was a mess.
Oh no.
I heard about that one later.
So is this a slurp situation?
Yeah, I think this is a slurp situation.
It's slurping time.
But let's
it
definitely
slurp o'clock?
zoom in and
Slurp
take a
o'clock.
look at it. You can keep zooming.
Oh, you want to look before
Yeah,
he
let's
grabs
look at
the
it
hose?
first
Okay.
while he gets everything turned on.
That's so cute.
Oh, it's really cute.
Oh.
I love that angle.
I think they're all open, so one.
Wow.
Sea star sitting on a heavily encrusted pillow basalt fragment. You can see it's tube feet.
Is that what you call those little tubes at the bottom? Little feet?
Yeah, those are tube feet.
Tube
That's how it moves
feet.
around.
2 feet.
Oh.
Oh, we got a rock in
Red
our...
palms. Not working.
Oh, there we go.
So yeah, what it's sitting on,
You want to do 7?
substrate-wise, that's a pillow basalt, and you
Oh.
can see the radial joints even through
For-
the crust.
Yeah, okay.
Oh,
Let's do
wow.
1. Start with 1.
Does that happen during like the cooling process?
That's part of the
or-
cooling process, yeah, and then this later
Okay.
broke apart,
We can zoom out.
exposing that structure. But that happened pretty quickly, given how thick the crust and bubbly the crusts look around here.
Yeah.
Tracked.
What was the word? Botryoidal? Did I say that right?
Yes.
Yes.
Nice.
I love a good botryoidal texture.
Me too.
Such a good word.
Almost as good as salacious ooze.
Ooh, salacious ooze
yes.
is pretty
That's
premium.
still my favorite, my number one. Okay,
Geology has
suction
a lot
on.
of good terminology.
I really want to have a geology party and have the juice be called salacious ooze.
We actually had some electrolyte drink last year
There it
that
Nicely
goes.
done.
the pilots called Silicious Ooze.
Did it,
I love
Is it
that
in
so
there?
much.
It was pretty good.
I didn't, there we go. It's in there.
All right. Nice work.
NA174-246. So that is a Goniasteridae sea star.
Ooh.
1.
Fantastic.
Amazing.
All right. We just took the sample just under halfway between waypoint 4 and waypoint 5 along this ridge that we're exploring.
Where did that? Okay.
Or the sea star.
We're about
Nope, we are not moving. Yep.
all right. All right, we do have a sample request, if possible, from our wonderful friend Asako Matsumoto for a snip of one of the Chrysogorgias if we see another one,
Okay. Yeah, we can do that.
since it's our deepest dive in the sanctuary.
I have a feeling we'll see another one.
I have a feeling we will see another one too. With associates, if we
There's
can.
one.
Ah.
Oh, perfect.
Just
Manifested.
on the right.
Yeah, if it has an associate, I think she may be
I don't know if
interested.
this one does,
Yeah.
but we'll have to zoom in and see.
Okay,
If not, we can go to the next one.
sure thing.
The right middle screen for the
Yep,
lasers.
right, but yep, right there.
Oh, perfect.
Those ones are notoriously hard to see.
There's something in there.
Yeah,
Yeah, maybe.
what's on here? There are associates. Yep, there's a Uroptychus in there.
Ooh.
Are they swimmers? Oh, that has another one of the.
And it has a
yeah.
tubular
Yeah,
on
yeah.
there. Yeah,
Ooh.
the hydrozoan.
Oh, a little bit of
And
everything.
another little pink something too.
Tortu?
You think you can slurp the whole colony? Zoom out a little bit. Oh, yeah, it might fit. Oh, well, let's try. They might. Well, the tubulator, I think they both would. Usually this Uroptychus holds on. Yeah. Like, I'll never let go. I don't know. I'm just afraid that it won't come all in the slurp. It might get stuck. It looks a little bit big to go through the tube. What do you think?
Yes, I think it's better to put
You
it
think
in
it'll go? Oh, let's try.
No. I don't know.
I ain't going down, going down, going down. Okay.
Zako says whole colony would be great,
Yeah.
if that makes things easier.
It's just a one, it should break. It's not like a black coral where it's like super bendy.
Okay. We're going for it.
And it's really, really thin.
Yeah.
Could we, if we're doing a whole colony sample, could we do a front bio box?
Well, I think we're going to try to slurp it all through.
Okay.
The question is whether the associates would hang around.
True, that's a good point.
The tubulator would hang out, but
the squats maybe
the
not.
squat might bail. Unlikely. Usually the whole thing comes, but it's kind of small, and you might squish it and it might swim away.
Okay.
Okay, try it.
Let's try the suction first.
Let's try
how
the
it
suction
goes.
first.
I think we only got one shot at it.
Yeah.
But it,
No, I agree. That's
there's
the way to go.
around, so if
Yeah, we can
if
always
we
try
botch
again.
It, we could
Yeah.
go to plan B on the next one.
Sounds good. I like plan A and plan B.
So we gotta go to the next jar, I guess.
Oh yeah, it's on flush right now.
All right. Jar. I know if I push the button, I'm going to hit the wrong way. Go. Yeah. Guaranteed.
Yeah. It's like the USB, you know?
Yeah.
You never get it on the first try. Even if you look at it and you're like, I know it's in the right orientation.
Sometimes you don't get it on the second try either.
I know. Yeah. It's that
Some people
superposition
speculated, yeah,
situation.
that they exactly.
All right. Suction coming on.
Can we zoom way in? Let me just touch the rock there so I know where I'm going. All right. All right, going in for the kill. There.
All right. All right.
Ooh, ooh. Oh my goodness.
Whoa.
Okay. Hopefully it'll...
What was this from?
That's afraid. That's what I was afraid would happen.
Oh no.
Ah, there we go.
Okay,
Yes.
okay.
All right.
Just a little
You get through
percussive.
that little bottleneck.
Success.
There we go.
Ah.
Beautiful.
Okay.
Nicely done.
Amazing.
Whew.
Love when plan A works.
Okay, plan
That was a good plan A.
Mm-hmm.
Some little red, is it a shrimp?
I think that's a shrimp, yeah.
Little shrimp friend.
Nematocarcinus.
Oh, how precious. I love it when they have colored eyes. They're so cool. Or is that reflecting?
28.
Whoa.
Everybody loves a good long-legged shrimp.
Mm-hmm. Legs for days.
Yeah, they just like walking. That's just what they'll do.
Looks good.
Sounds like they need some boots if that's the case.
Yeah, they need some. I just imagine little boots on this shrimp.
Boots that were
Shrimp
made for
boots.
walking.
Well, it's the urchins that have the boots.
True.
Oh. Have y'all seen the aquarists that put little cowboy hats on their pet urchins? It's so cute.
Which one?
How's our comps doing? Give it a look about.
Let's see. Rest looks good, main looks good, motor looks good,
Right.
craft looks good, term looks good, thrust looks good.
It was just a couple of drops, so.
Just a little bit. And probably because we were running everything.
So we're seeing a little
Okay.
change in the substrate here.
Onward.
Onward. Let's go.
Yeah,
Got some good stuff.
yeah. We're seeing some substrate shifts here. Still lots of pillow basalts. Still lots of debris, but none of this debris seems to be particularly accessible. And now we're back into what appears to be in-place lavas intercalated with some debris, and these ferromanganese crusts are very thick and quite well developed, which means it's been like this for a really long time. But you can also see that there are some areas where there's more parallel striations in the crust instead of just that straight up botryoidal texture,
Faster.
which may or may not have something to do with formation in a current. Not totally sure, but
Okay, I'm gonna drive by.
that's how I've been interpreting it.
Doing a drive-by.
There's more of those feeding traces here that we've been mentioning here and there. Probably not that crinoid doing them, though. I would be very surprised by that. Yeah, the geology is kind of rapid firing some changes here.
What do you think caused those changes?
I'm not too sure. I think that might be more the erosional, some of the early erosional and like collapse, like slope instability stuff
There's
that
a
I was talking
little-
about earlier.
Something on the rock right there. The loose rock right lower right.
Lower right. I'm not seeing it yet, but I'm
It
looking.
just went out of frame.
Ah, okay.
But there's like a boulder with a little clear something on it.
Oh,
Oh,
yeah,
little
that
puka
one.
No,
here.
further right.
It's like on the, it's like just out of frame. Like right there.
There's like a
right
little
there.
Oh,
circle.
yeah,
You see
right
the
there.
circle?
Yeah.
Something kind of spongy, maybe?
Well, let's
Or
Or
find
a
out.
tunicate?
Oh, I think it might be a tunicate.
Yeah, because it's kind of ringing a
Look at
bell. It looks a little like
Yeah,
that
see
tunicate
little
from earlier.
ring?
Ooh.
Yeah, it looks like a
Big
sea
mouth.
cucumber's been here too.
You said come wide?
It's in the dirt.
I think it's on the rock. It's just really see-through.
Yeah.
Boop. Yep, definitely on the rock.
Oh,
It
yeah.
wiggled when you touched it.
Yeah.
Do they
That
have
looks
like
like
an,
Your
sea
big
anchor?
mouth.
cucumber.
Yeah, the Megamouth Tunicate.
do they have like an anchor to it? Or, oh, it is? Oh, wow, it's so
Yeah, so
see-through.
you
I
could-
couldn't even see the base there.
Yeah, you can see
Yeah.
the base right there.
Whoa.
Yeah, and it closed its mouth when we moved its house.
So I didn't like that.
I don't like it when my house moves either.
Yeah.
Cool.
How neat.
Very long.
Another Chrysogorgia. With the Uroptychus. It's a cutie.
Wow. I can't get over how pretty they always are. They do remind me of cherry blossoms.
They do.
Red cotton candy.
I'm pretty sure it wouldn't taste like how it looks.
No.
A little crunchier.
Some umami maybe.
I think seasoning can fix.
So what would be your recommended seasoning?
I think
Sugar.
sugar and butter, maybe some cinnamon.
Okay, we're making it a dessert.
I think so.
All right.
Yeah.
Yep.
Man, we're coming up on 250 samples this expedition.
Wow.
Oh, wow.
That's a lot.
250.
Most of them are water.
A lot of them are water.
Gotta get that
Speaking of
ED
which, we haven't taken any Niskins, but there's been not
No.
much reason to. So we are currently about 2/3 of the way between waypoints 4 and 5 on our 14 waypoint dive. So we're actually making better progress, like faster progress than we had initially expected on this one.
Yeah, no, we're doing great.
We are doing awesome.
I want to see how much we've covered. We've gone, let's see, about 165 in that direction. And about 300 in that direction.
Man, that's
Wow.
booking it in ROV scales.
Yeah.
Yeah,
Dang.
we've covered quite a bit during
Yeah.
our watch.
And once we get past waypoint 5, this ridge will widen a little bit. It's going to be like a little step area that we're getting into, I think. And then after waypoint 6, we should start seeing a little bit of slope again. So we'll see if we make it that far by the end of the watch. No idea whether we will, but you'll see some morphological changes for sure as we move into the wee hours of the night. Either with us at the end of our watch or with the 12 to 4 watch. But right now we're looking at a big pile of pillow basalts, which is my favorite.
I really want to see an octopus.
Yeah.
Me
Oh,
too. We have not seen
yes.
any this
Oh,
expedition
I
yet, have
know.
we?
No.
Usually we get like one Dumbo an expedition if we're
Yeah.
lucky, and they're just cool.
Steve was really hoping we'd see another Casper octopus.
Yeah,
That was the
Oh.
best.
we saw one of those a couple years ago. They're very, very cute.
They're super cute. We don't know what it is. It's a new species.
Oh, wow.
What's the likelihood of an octopus like appearing in this depth?
I mean, it's It happens quite frequently at these depths. It's just hasn't happened yet.
Interesting.
I'd say that it's like 50-50.
Well, do they prefer to be in an area that has more coral or other kind of abundance, or?
I don't know.
Yeah.
It's a great question. We would have to do some analysis of observations to see if there's any correlations between octopus observations and, you know, community densities. But, you know, I can speculate, but I'm not sure.
That is a really interesting question.
Huh. I love questions that lead to more questions.
Oh, yeah.
Oh, we have so many of those
Yeah.
around here. Because, yeah, we may not be seeing a whole lot besides rocks around here, but there's a whole lot that we won't see because it's running away from the ROV.
Yeah.
Oh, yeah.
We're friendly most of the time.
Or if
But
you're a
we're
fish,
also
you're hanging
really
out behind
loud.
somewhere. Yeah. Yeah, like we can definitely hear it on deck when the high voltage gets turned on with Herc.
Yep, goes.
And that carries in the water
Yeah.
way further. So we're just like, just blasting sound.
Kind of incredible that we ever see fish ever then.
Yeah.
Yeah.
I want to see a nice big Caulophacus.
What is that?
That's the rat tail fish.
Oh, the rat tail.
Ooh, yeah.
Okay. I need to get better at my scientific names.
I mean, you're pretty good for a geologist, I have to say.
Well, thank you. I'm flattered.
Oh.
I don't get many places to practice that off the ship.
Right? Yeah. Like, when would you see these things unless you're sitting back there in the science row?
When I'm working short team for the Okeanos.
Right.
So we had a good question from one of our guests asking what depth we are at. So we are at 2,785 meters for anyone who is curious how deep we are. We have a steady kind of incline to like a more shallow depth as we go, Val, for this one. Or as we reach the peak.
It's going to be relatively flat, a
Relatively
little bit of an
flat.
incline
Just a little
for quite a while to go. And then
Yeah.
you see here after a waypoint 6, the contours
Yeah.
get tight again.
Mhm.
Yeah.
Okay.
It won't be as steep as before, though.
Yeah, that was really steep. Oh my gosh.
It was a good place to look for talus.
All right. Welcome to anyone just tuning in. See, we have a lot of new guests from all over the world. Give y'all a shout out real quick. We got fans from Belgium, Czech Republic, Estonia, Spain, France, Guam, Marshall Islands, New Zealand, Philippines, Portugal, Russia, Australia, Germany, United Kingdom, and the United States. Welcome everybody for tuning in. Again, if you are streaming through YouTube, I'm going to recommend y'all go to nautiluslive.org. If you want to ask us any questions, our chat is live and open. My name is Giselle De Leon. I am a science communication fellow here on board the EV Nautilus. When I am not on board here, I am a lead STEM educator at a museum in San Antonio, Texas. Anyone else want to introduce themselves for anyone just tuning in?
Oops, sorry, I'm
No, you're
typing.
good. You good.
Maybe I can start and then we'll go around
Yeah,
and finish
ping pong.
with Val. Okay.
Yeah, sounds good.
So I'm Luisa Dueñas. I'm from the National University of Colombia. I'm based in Bogota, and I'm here also with Ocean Census. I'm sitting in the Science 2 seat, and I'm deep sea biologist helping with all the biological samples we're gathering in the expedition.
Hello. My name is Hovil Dunkel. I'm from the Solomons. I'm attending the University of the South Pacific. I'm an ocean science intern serving as a data logger. Thank you. Oh, yeah.
Yeah.
Phil's having a good time back here.
Yeah.
In your front row, I'm Megan Putz. I'm the navigator on this watch, and I really like deep sea animals, and I'm going to tell you all about them.
I'm Robert Waters. I'm sitting in the Herc seat, and I don't know, passing on.
Hi, I'm Levi's. I'm on the Atalanta seat. I'm the ROV engineering intern.
He's the ocean.
Oh my gosh.
He wants to eat the ocean.
And I'm from Guam. Yeah.
Hi, I'm Amber. I am the video engineer.
Little something on the seafloor.
Oh. Val, do you want to introduce yourself just like real
Bitty
quick?
thing there?
Yeah, it went right behind the rock. There's like a right over here. And it's right behind the rock right there.
Oh, you gotta spin around?
No, you just have to like move forward because the rock is blocking the thingy.
Oh, I see. I see.
Yeah.
Oh, wait, where's the basket?
Is
We're kind of
cucumber?
catty off
I
us
see
What
it.
is
Yeah,
that?
I turned the camera
here.
to point where I wanted you to go.
Oh, no.
I saw.
Ooh.
I swear I just saw a polyp way off to the left too, but it was in the water column.
Oh, it's gone now.
Yeah, it's gone now.
Those are hard to catch.
They move fast.
Yeah, there's a little synallactid sea cucumber.
It's a little one.
It's a tiny,
It's so
tiny little.
precious.
Would this be a juvenile? Or,
I don't know.
yeah.
Unknown. I mean, it's small, but it could be an adult. Or it could be a juvenile. We don't know these very well. A lot of them look the same.
Yeah.
You know, they're just kind of a nondescript, kind of pinkish purple blob on the seafloor.
Yeah.
It looks like a gummy.
It does look like a gummy.
It does. You're not wrong. It doesn't taste like one, though.
No, definitely
But
not.
those are, like, people eat them, yes?
I've heard of people eating them, yeah.
Have you ever seen the true facts about the sea pig?
No, I haven't.
Okay, well, you should look that up.
Oh my gosh, I
But
think I will.
it tells you true facts about the sea pig, which is a sea cucumber.
Oh,
The family Elpidiidae.
wait, no, I have seen this. Oh my gosh, I love those series.
Yeah.
True facts.
True facts. One of the facts is bacon from a land pig is delicious. Bacon from a sea pig. Maybe not so much.
Sounds like a true fact I can get behind.
So what I'm hearing is someone tried it.
No, I think
Yes.
they are, like, part of a normal cuisine. I could be very wrong, though.
I could have sworn I've seen like somebody being like, yeah, there's cans of like sea cucumber that you can
Oh,
eat.
they do eat sea cucumber. It's usually the shallow water ones.
Yeah, shallow
Ah.
water ones.
Um,
They're awful.
Not the deep sea ones. Those
really?
would end up being really
yeah.
expensive because how are you going to get them? Can't just
Yeah.
dredge for sea cucumbers.
This is true.
Well, Levi, lucky for you, a member of the back row has actually tried a sea cucumber and does not
Yes.
recommend.
No, it does not.
Oh.
Chinese restaurant, don't try it ever.
What did
Yeah, I
it
think
taste
that's
like?
one of those things I just couldn't do.
No.
Just bad.
The texture, it's like, no.
Yeah, I'm very sensitive to texture of food, and that sounds like something I would have a lot of trouble with.
Have you guys tried sea cucumber with Kool-Aid?
With
No.
Kool-Aid?
Yeah.
What?
They serve it
tried
at Palau. Yeah.
Ah.
It's good.
Really?
Yeah.
In San Antonio, we have pickles with Kool-Aid.
Oh, pickles?
Yeah.
Those are good too. Trust me, those are good.
That makes more sense.
But why Kool-Aid?
I'm still surprised on the fact that a sea cucumber is known as a sea pig. Is
Well,
it?
Some of them
So,
are.
no, so
some of them.
only ones that with
Oh.
the legs that walk around are the sea pig. Yeah, the scotoplanes, those are the sea pigs.
So,
Oh.
Belle, there is one that is very famously known as the headless chicken monster.
Yes.
Yeah,
I'm
that's
serious.
Ipniastes.
Yeah.
No
yeah,
way.
The
Damn.
Ipniastes is the headless chicken monster.
He's looking it up. He doesn't believe me.
Yeah,
No, I'm trying to look what it looks like.
yeah.
They look like
They're really
They're
cute.
A headless
Purple.
chicken.
headless chicken monster.
And they just kind of flap
It's-
around.
It's a really accurate description, except that they are purple.
And they do crunches as they swim.
They
Yeah.
do, yeah.
Do they taste like chicken?
No.
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 You see a lot of them swimming around on the abyssal plain when, like, we're doing shipwreck dives.
Well, we saw a number of them in the Atalanta view
Yeah.
earlier in the dive. At first I thought it was like something cool, like a fish, but then it was just a sea cucumber. Not that sea cucumbers aren't cool, it's just...
Are we passing notes in the back row now?
Guys are getting silly back there.
What's going on?
Okay.
Of course they did. All right.
All right, back to the
I mean,
science.
I guess they're not wrong.
We got some giggle
They're
bugs.
not wrong. Back to the science.
Back to the science, back to the science. It's fantastic.
So speaking of rocks,
Yeah.
I think I might like to pick up a rock here. We've gone up quite a bit.
Okay.
We've gone quite a ways laterally, so this might be a
I
good place
see
to
a
grab
nice
if
one
the
right
ship
over
is
here on
not
the right.
moving.
Ooh, the darkish one?
Are you looking at that one? Because
Yeah.
it might be a little
That's
big, but we
huge.
can go check it out.
Is that big?
I think
Okay.
so.
Yeah, that looks pretty big.
It just
Maybe.
had a really
Huge.
nice shape.
It does have a really nice shape.
The lasers were really far away, so I
Very
thought they were...
pointy.
That's a,
You're having the same problem I was having on the last
it's a
collection.
massive rock. We can't
Yeah.
get that right.
Oh, okay.
I want to make sure we're
Dang.
managing our ballast since we're going to be down for a while. So,
Yeah.
yeah, I'm after the smaller ones.
It looks like there's a number of good options.
This is another problem where it's too many good options, you know?
Yeah.
Yeah.
I have this
Oh,
problem in
man.
parking lots.
Oh, yeah. Yup.
So you understand my pain right now.
Oh my god, there's
Like-
too many parking spaces. Like, where should I park?
Where are you parking in Hawaii, by the way, where there's enough open parking spaces to have that problem?
Late at night.
Ah. Oh, yeah, true. That's a good point.
Never at Costco.
Let's see. Could we just park, like, I don't know, and take a look at these right here? I think this is as good a place as any.
Sounds good.
Yes, I am just making decisions now because I don't want to hover here forever. Okay, all right. Looking around, looking around. Let's see what looks reachable here and is also about 15 centimeters. Ah, there's the lasers. There's my reference point.
Stupid
Could we
chair.
check
Yeah.
this one
It's
here? It
such
might be a
hard
little
chair
small.
to move.
Yeah. We cheaped out on the chair.
You should never
All
cheap
this
out on the
stuff,
chair.
And then you cheap out on the chair.
Yeah, like these chairs are expensive.
I was gonna say, the
those are
Herman Miller chairs.
Millers.
I know,
Cost a pretty
really
penny.
expensive.
$1,500
I know,
chair.
it's crazy.
This is like a $400 chair.
I know, like, all of us except for you get like an expensive chair.
Believe me, I complain.
I'm sitting in a chair that has one arm.
Yeah.
My chair
well,
has the whole.
it still cost $1,500 when it was new.
It is nicer than my chair at work, though.
Oh, yeah, track
I
back.
think-
These chairs have been around for quite a while.
Yeah.
Yeah.
Probably 20-year-old chairs.
Like, old chairs on a floor that moves around a lot, you're doing pretty well.
Yeah.
So I can't knock it, but it is kind of funny.
You just start poking it?
Yeah.
Poking some things?
Maybe we poke that one, just for giggles.
Just for giggles. Uh-oh.
Oh,
Ooh.
it moved.
Oh, I like the angularity on that one. Oh yeah, that looks nice.
All right.
Oh, yes. That is a
Yeah.
definite keeper.
Okay.
It's so hard to make a decision.
I think you did.
I made a decision. It was not easy, but I made it. There's just too many good choices. And
Maybe
it's,
shift sample mode.
yeah, I think D is open, correct?
Yeah.
Yeah.
There
You
we
say
go.
E?
D.
D.
D. The one nobody goes for.
It's to give you a nice challenge.
No problem.
Perfect. Easy. You made it look so
You made it look
easy.
very easy. Thank you. I mean, after some of those earlier dives where we were just looking for rocks for hours, it's just like, what do I do? They're everywhere.
Yeah.
Yeah, true. There's some dives you didn't even get any rocks.
Hmm?
Weren't there some dives where there were no rocks collected?
The first dive we got one rock, but it was a good rock.
Yes, it
Ooh,
was.
That's a good one.
I think we managed to get rocks every dive. We've gotten, there have been a couple where we ended up with no lavas, but one of those did give us a sandstone that my collaborator is going to take a closer look at, and we're going to see if maybe we can get an age date from that,
Oh, how exciting.
if it has the right material in it.
Do you know the process on how it differs from your process to aging and deciphering with that?
He's the one who does all the
Oh,
age
okay, all right.
determinations. I don't do that in my lab.
Yeah.
I do stuff with a lot of dangerous things, but I'm not the one who bombards samples with neutrons the way that he does in order to do that. So
Oh, wow.
yeah, we have
That's
two
cool.
two completely different techniques that we do for lab prep.
Wow.
So he's an expert in that. I'm an expert in the other part of it, and combining those two things, we're basically unstoppable. It's great.
I was going to say, that just sounds amazing. Wow, it's very flat.
Yeah, we're getting into something now that looks more like a sheet flow than a pillow basalt. So this is more lava that came out all at once, and instead of kind of worming its way around, it's a much more extensive kind of broad lava flow.
So would this be a slower process than the
That is
pillows
a
you were saying?
Caulophacus.
Probably a little faster. It's more voluminous in eruption for sure, but as far as eruption rate, that depends on a few different
the
factors.
Caulophacus.
Oh.
Pillow lavas tend to be pretty slow moving though.
Oh, I think this is a winner. You see
Oh,
this?
yeah?
You see this little thing on
Yeah.
the Caulophacus?
Oh, that's one of our, okay.
This is a Steve request. We want to
the
collect
ship
this.
is stopped, right?
I'm gonna stop the ship.
Okay.
What is it?
Ah,
Bridge now.
it is a limpet.
Oh!
Can we hold position?
How cool.
Thank you. So this is actually an Aplacophoran. It is a type of mollusk that has no shell.
Oh, okay, not a limpet.
Oh, what?
It just
Oh.
looks like a limpet.
Oh, it
Yeah.
does look like a limpet, though.
It's
Listen
a
to
really
Megan, not
weird
me.
thing.
It's a very cool looking thing.
It's a very
though.
cool thing. This is probably a new species.
Okay.
Oh,
Yeah.
wow.
Okay.
So we're going to
Definite
suck
collection.
this off the Caulophacus.
Okay. How cool.
Yeah.
Wow.
Oh, it's
Steve's
kind
gonna
of
be
a
so
big one.
happy.
Steve's gonna be really happy.
Yes.
Are we close enough?
Yeah, I think we need to probably scooch a little closer. Just suction
Yeah.
it.
A little bit.
Wow. And do we know what kind of role that plays? Is it just an associate on a sponge, or what is it? What does it do?
What's it do?
Yeah, like what is its
Um,
job?
Well, they could be sponge predators.
Oh.
All right, this is a. Can you switch over to the jar cam?
Yep.
And we often see aplacophorans and selenogastries associated with sponges and corals. And the selenogastries are very similar to aplacophora, and they are known coral predators.
Oh.
But this is a really cool observation.
That is. That was an awesome spot.
Yeah.
And that one's actually kind of big. You know, it's like maybe like 3 centimeters, but it's kind of big for one of these.
So the more typical size, what do they tend to be?
Like 1.5.
Okay.
Oh,
Yeah,
wow.
that's a pretty big
massive.
size difference.
And are they?
But we found other ones of this type of animal in inside sponges and
Okay.
in sponge collections before.
Oh, wow.
But this one looks different than what has been collected, and definitely larger.
And does it have the ability to swim away, or they usually
Alright.
kind of stay?
Suction coming on. Oh, there it goes.
Oh, wow.
Oh, excellent.
Down in the jar.
There it is.
Oh.
Perfect. That was fantastic.
Oh, amazing.
Oh, maybe it was 2 of them. Oh, that's wild.
Oh, did you want to look again?
No, in the jar.
Yeah.
Once it went
you
in,
want to look in the jar?
yeah, in the jar.
Yeah.
It looks like there was one maybe on top of each other. Maybe they were
something's
mating.
going on.
Oh.
That's
Okay. that's something to make note of in the log.
Yeah.
Wow. Okay. That, if so, that's even more unusual.
Yeah, that is cool.
If we've got two of the same species, different sex.
Oh, wow. Incredible.
Huh.
Wow, what luck to stumble upon that.
All right, and that was number 249.
249.
249.
Next one's 250.
Oh man. Ooh, what's the sample gonna be? Is it gonna be bio? Is it gonna be water?
Water.
It's gonna be water. Luis is on it.
Water.
Hey Amber, it looks like you made the sit rep.
What did I do?
You were featured in an awesome picture. That's what you did.
Oh, okay.
Oh, a new kind of sponge to look at.
Which one?
Sure enough.
The straight ahead.
Oh, that's just sediment over there.
Yeah, it's just a little sediment pocket. There's actually 2 of them. I think they're both polyopagons.
Ooh.
So this is making me increasingly curious whether things are going to pick up a little bit once the slope gets steeper again.
Ooh.
One polyopogon. Probably. I think things
Yeah.
are going to start picking up. We're starting to get into that depth range where
There's something
things
back here
get dense
too.
again. Okay, we're good on these.
Seen enough?
Yeah.
Yeah. Yeah, so if things start really picking up, I'll be on the lookout for any potential good transect spots.
Arms hanging out in the breeze.
And what would be the kind of the qualities that you're looking for when you're going to
They
start a
turned it off.
transect?
Oh, yeah, don't do that.
Well, we actually have a fix, though.
Yeah.
You can let it droop, and then you bring it up, and it stays with the button off.
Is that
Oh.
something moving over there?
Want to see?
What is
Yeah.
that?
So you
if
hit
it
the
goes-
you hit the blue
see that?
button,
The dot?
and then it goes bloop.
So it had already blooped.
It already blooped once.
It blooped,
So-
and then I can bring it up,
Up.
and then I can let it off. and it'll
Okay, is
stay
it
there.
like a pressure release
Yeah,
situation?
there's a pressure thing going on.
Oh.
So if you let it droop all the way till it stops, and then you bring it up, and then you can release the blue button, and it'll stay.
I
But if you
see.
try and catch it as it's falling,
Right.
you
Then-
can
then
never,
it
you'll
just
never-
always kinda
it'll
keeps
just always
going.
fall.
I see.
Yeah, I reported
That's
that
a
to the.
weird quirk.
I reported it to crafts. Said this is what's going on.
Yeah.
Yeah. So they're working on it.
Yeah, so what we're looking for with these transects, to answer your question, Giselle, is a space that we can document for us at distance, the diversity and population density that we see along an area. And we've been trying to target some relatively flat areas, but we can also do this on sloped areas. And we've had some pretty good success on recent dives. Doing some of these transects along slopes. So we've been finding higher population densities there. And what we're using these for, this is something that Fanny is interested in working on out here. She constructs photogrammetry transects from the imagery, and she can use those to quantitatively study what these communities are doing along these transects.
Ooh, I hope we get a good one.
Yep. So yeah, we set up a 50-meter line, and we just fly Herc along that without stopping, without changing the zoom on the camera, and just letting it fly forward a couple meters above the seafloor in order to capture the image properly.
How cool.
So it's time to change the pumps in the multipuffer.
Roger
Okay.
that.
Got it.
Oh, it's a sleepy looking star. Let's look at this star.
Oh, do we need to stop and take a look?
Yeah, we're just gonna zoom in.
It does
Okay.
look
I think it just
sleepy.
might be a sleepy brisingiid.
Yes.
It looks
Yeah.
so sleepy.
It's
You don't
sleepy.
usually see them as sleepy, so.
Yeah, well, when they're not eating, they rest their arms.
Oh,
I do
they're-
the same thing.
Right?
Yeah.
You don't just keep your arms in the air trying to catch food,
I don't know,
all
that seems
day.
like a good idea.
right? You're like, oh.
Okay, so pump 3 is off and pump 5 is on.
3 is off, 5 is on.
yeah.
So this area where we see different coloration on the arm, could that be new growth that's lighter?
Probably, I think you're right. Peas. Bridge now. Hello. Can we make another 30-meter move? 310. Thank you.
There's some more dead sponge stalks we're kind of seeing in the frame of it.
Yeah, lots
Yeah.
of dead sponge stalks. That probably means there's really cool live sponges up top.
I hope so.
Me too. I love a good surprise.
Alright, so we're just barely past waypoint 5. And the slope will start slowly picking up as we approach waypoint 6, and it'll get steep again after that as we head toward waypoint 7. And we're in the last hour of our night watch. So this has been flying by.
Yeah.
And we found some cool things.
We have.
Yeah. Ooh.
It
So
might be
excited.
sparse, but they were cool.
Got that quality. High
Yeah,
Yeah,
quality.
this is one of the challenges of seafloor work is it's a little bit more sparse on these really deep dives. Take a little longer to get down to and survey, and maybe a sponge there. But since we don't look at these as much, yeah, we're probably looking at a lot of newer things or like range extensions that we wouldn't otherwise have known
Something
about.
There's a polykeet right here. See the little wiggly?
Oh yeah. Yeah, that's one of the swimmers I was seeing earlier.
Yeah.
Oh, that's a big one.
Oh, whoa, look at that. It's so cute. Doing a little dance.
Oh, it's landing.
So as much as I'm not fond of house centipedes, although I know they're good, I just don't like the way they move. The movement of the polykeets reminds me a lot of how house centipedes move around when they're running.
Yeah.
Although I'm fine with the polykeets, but that's probably because